Hi all, Dr. Clark here again. Today we're going to start with meiosis. And so in the previous lectures we were talking about mitosis. Remember, that's nuclear division in somatic cells. Today we're going to talk about meiosis or meiosis, and that is nuclear division in germline cells or reproductive cells, and that's it. It's just in the reproductive cells. So, you can see some things that might be fairly familiar, like metaphase. Here's a cell, a germline cell that's in metaphase. Here's a germline cell that's in anaphase. And you can see these same kind of phases, same kind of processes in meiosis and mitosis. There's just a little bit of difference, and that's what we're going to discuss mainly in the next few lectures. Okay, so to discover this, it's interesting. We can kind of give a little bit of history of this. So even when Darwin came up with the theory of evolution by means of natural selection, we did not know how parents passed on information to offspring. We knew that the offspring looked like their parents, at least had some characteristics of their parents, but we didn't know how that occurred. And Darwin came up with his own hypothesis, which he called the genual hypothesis, which was that every organ and every piece of the man and every piece of the woman, just a small, minute piece of them, would break off and it would all go down to the reproductive parts, and then they'd pass it on, and then they would mix together and form the new individual. Sounds a little bit crazy, but we didn't know there was such thing as DNA, so it was a fairly good guess at the time. Mendel was working on his research on genetics in what's now called Russia, but he was working there in a monastery. He was a monk, and he didn't know either, and had his own kind of ideas. He just called them characteristics and instead of genes and all this. And we'll get to Mendel in a little bit, but again, had no clue. So it wasn't until really the 1900s, so like early 1900s, that we found out how gametes and how reproduction actually works. Okay, so regardless of the organism that we're talking about, whether we're talking about plants or animals, or we're talking about protista or fungi, if they can do sexual reproduction, they produce gametes. And gametes are the reproductive cells. Now, they can be a huge variety. It could be the normal stuff that we normally talk about, like eggs and sperm. It could be pollen and ova, spore cyst and ovocyte. There's lots and lots of varieties of what this might be. Okay? But as long as you're recombining to restore the normal chromosome count, it's considered sexual reproduction. And we'll look at some examples in a little bit. So gametes will fuse. So sperm, egg, pollen, ova, etc. They'll fuse to form what we call a zygote. And so the gametes are half of the normal chromosomes. So if the individual is a diploid organism, then that individual's sperm or egg or pollen or ova or whatever you want to call it is going to be a haploid. It's going to have half the number of chromosomes. Then when the two haploid cells, sperm and egg, get together, it restores the diploid number if the organism is diploid. If it's a tetraploid, meaning they have 4 copies of every chromosome, their sperm has 2 copies, so it's a diploid. Their egg has 2 copies, it's a diploid. Returns the tetraploid number. Okay? Now that formation, that fusion of the gametes is called, the result is called a zygote. Okay? We know what the fusion is called. We normally call it fertilization. And there's lots of other terms for this: pollination, syngamy. There's lots of things, and it's really just the fusion of gametes. Now, if you think about what would occur if instead of meiosis occurring, we had mitosis. So let's say we have sperm that has 46 chromosomes and egg that has 46 chromosomes. If we're talking about humans, and they come together, now you have 92. Well, then let's say that individual, the offspring of that individual, that zygote, grows up and reproduces on its own. Now that individual would have 92 chromosomes in their sperm, and they might mate with someone with 92, and it would just be additive. And so by the time we rolled around 4.2 million years later, the Homo line got to Homo sapiens, us, yeah, we would have hundreds of thousands of chromosomes, if not millions of chromosomes. So meiosis has to occur in order to maintain the normal chromosome number. Okay? So you have to have a way in which you can half the chromosomes so you don't get an additive chromosome method. So just what I said before, with each fertilization, no matter how many chromosomes you had, you would double the chromosomes if you don't have a way or process by which you can half the number of chromosomes. Meiosis or meiosis is the process that does this for us. We normally call meiosis a reduction division. So you're reducing the number of chromosomes in a given cell by half. So if there's 46, then the gamete is going to have 23. If there's 64, the gamete is going to have 32, and on and on and on. So it really makes no difference what organism we're talking about. There's lots of organisms out there. There's lots of organisms that have very different numbers of chromosomes, but the process of meiosis is going to divide their chromosomes in half. So it can return the original number when the zygote is formed. And most of us know something about this process. We know what fertilization is about. Sperm, egg, return the diploid. So the egg's a haploid, if we're talking about humans. The sperm's a haploid. It's showing 3 chromosomes here, but you would know that you would actually have 23 chromosomes. And 23 chromosomes in here, and then 46 chromosomes here. Because you're going to have a pair of each chromosome type. So you'd have 1 through 23 in the sperm, 1 through 23 in the egg, and then you would have 2 copies of chromosome number 1, 2, 3, all the way to 23 in that zygote. That's where the diploid comes. It's 2 copies of every chromosome. Now, like I said before, we could change that to tetraploid, then this has to be diploid and this has to be diploid, and on and on and on and on. Okay? But for the most part, we'll talk about diploid parents just because that's what we are, and it's kind of easier to understand for most people when we work with smaller numbers. So the zygote is diploid, and so is the germ cells before they go through meiosis. They're diploid. So the gametes themselves would go through meiosis and become haploid, and then return the diploid number. I hope that's pretty self-explanatory. Now we know that some organisms have the capability of doing asexual reproduction. Okay? Asexual reproduction is, for all intensive purpose, mitosis. Okay? Not meiosis, but mitosis. You're just taking one cell and you're copying its DNA, dividing it into another cell. And that just happens over and over and over again. And that's how asexual reproduction works. Now, in some cases, it's a lot more complex. In some cases, there's behavior. In some cases, like parthenogenesis, where there's no males. Females will entice other females to produce eggs and then fertilize those eggs themselves, or cause those eggs to mature themselves, and on and on and on. And it's not just a prokaryote system. Okay? There's lots of eukaryotes that go through asexual reproduction. Now, prokaryotes do it most often, and we already talked about this. Remember binary fission? So bacteria copy their DNA, and they stretch out, pinch off, make 2 new bacteria. Okay, archase same way. But asexual reproduction occurs in eukaryotes also, and you can have some organisms that can do both. For example, you just take things like strawberries. Okay? Sexually reproduction, because you see a flower, and that flower is an attractive body part to the plant. Okay? So that flower is to attract things to come in and pollinate them. The pollen is the sperm, and then the pistil or the oviduct, ovaries are the female parts. And so again, the pollen has to hit the top of the pistil, then a pollen tube comes by, and the pollen goes in and fertilizes an ova to form seeds. Now, so strawberries do this, and you notice this because when you pick up a strawberry to eat it, the seeds are on the outside of the strawberry, little teeny tiny seeds all over the outside of the strawberry. So they do have sexual reproduction. But they also have asexual reproduction. For those individuals who've ever grown a strawberry, you'll see that they send out runners. Okay? These long runners, sometimes they go underground called rhizomes, but sometimes they just run across the surface, and then they'll set root and produce a new plant with flowers and produce strawberries. Okay? But it's all the same individual. It was just going through asexual reproduction. Okay? And there's tons of plants that do this. Okay? A common one in the western United States is aspen trees. So if you see like a hillside with a bunch of aspen, more often than not, that's only maybe a couple individuals. And so they just keep sending runners out, new individuals pop up, okay, but it's all the result of maybe a couple individuals occupying that hillside at one time, and then they just reproduce asexually. And so lots of plants have the capability of doing both asexual reproduction and sexual reproduction. Now there's other eukaryotes out there that can do asexual reproduction. There's a species of lizard that occurs in Colorado, Idaho, and Washington. It's called a whiptail lizard, and there are no males. Right? It's just females. The entire species, there's only females in all the populations. And like I said before, behaviorally, there will be females that act like a male. So do a little dance kind of thing, and the female will get excited and fertilize herself, right? And then that female then becomes a male and entices the other female to go through asexual reproduction also. And so there's interesting situations when it comes to asexual reproduction and parthenogenesis. And then there's a whole other group of sexual, asexual patterns that we see in other organisms, from hermaphrodites that can fertilize themselves to hermaphrodites that can't fertilize themselves. But so sexual reproduction, asexual reproduction, it's variable across the animal kingdom and the plant kingdom and fungi and really protozoans and all kinds of things. There's lots and lots of varieties to it. And so we'll talk more about that near the end of this lecture series. So as far as sexual life cycle goes, it's also varied. So we normally think about this one here, where the animal itself is the diploid version. Right? So it has either 2 copies of the chromosome, or maybe they're more than diploid. Maybe they're tetraploid or hexaploid or octoploid. So strawberries are octoploids. They have 8 copies of every chromosome. So we have variable things, but normally we think of the adult having the normal set of chromosomes and their gametes having half that amount, okay? Just like this system. So sperm and egg produced. Okay? But there are other organisms like lot of protista where the diploid version, or the most chromosomes, are actually found after fertilization. And then they go into meiosis, and so the living form, the form that you would actually see, is like sperm or egg swimming around, and they spend most of their time in that form and very little in the fertilized form. And then you can have things like in, ferns and things like that, where they have kind of two adult forms. They have a haploid adult individual and a diploid adult individual. And so these are some of the prehistoric plants that we see, ferns and things like that, and some algae that have been around for a long time. They have this kind of not quite one way or the other system, and can be fairly interesting to look at and study. But I'm just showing you this so you understand that not everything is simple as we produce it, and it's not like us. Okay? Most organisms on the planet are not like us. They don't do things like humans. So I try to present as often as I can the different variations of things, so you understand that yes, most of the textbook examples and things like that are all based on human body and human experiences, but not all organisms are that way. So in animals, for the most part, we'll talk about animals that are going to do sexual reproduction. So from here on out, we're talking about animals and plants and other organisms that are going through meiosis. Okay? And so we're not going to talk about asexual reproduction anymore. That's mitosis, and we already covered that. So now we're talking about the formation of gametes. And I told you before, meiosis happens in the germline cells. Okay? So in the germ cells, and that's it. It's not happening in somatic cells. So just germ cells. But the germ cells are like somatic cells, and they're diploid, or whatever the normal chromosome number is for that organism. And then the germline cells become differentiated into whatever that organism is. If it's a female, it goes into like ova or ovaries, or whatever it might be, eggs. And then if it's a male, it goes into sperm, pollen, something along those lines. And so that's the germline cell. But in order for that to occur, you have to go through meiosis. You have to produce half the original number. If we're talking about diploids, you have to produce haploid gametes. So pretty simple. Okay? You can see here, we know this piece really well. Okay? Adults, sometimes not so much adults, but get together, pass sperm, egg, okay, and produce diploid individual. Haploid from male, haploid from female, diploid zygote. Now, for a good period of the zygote's formation, it's only going through mitosis. Okay? Then eventually, through this mitosis process, there will be meiosis going on in that individual. And I want you to think about that a little bit. The majority of the organism is going through mitosis, but then there's a triggering point, and meiosis kicks in. And you should be saying to yourself, puberty. And whatever puberty is, whatever maturation is for an organism, that's the point at which those germline cells are triggered to go through meiosis and start producing gametes. Okay? We do this, well, for guys, for males in the United States. So I'm going to give you United States statistics, not worldwide, because the United States is a little bit different. In the United States, for males, maturation, puberty happens somewhere around between 11 and 12. Okay? For females, it's closer to 10. Okay? And this has been decreasing over the last 20, 30 years. We think it has to do, most people think it has to do with high fat content in our diet. and too many calories. So it's linked, or at least shows similar patterns to obesity in the United States, with the same thing happening as our children become more and more obese. They mature earlier and earlier in age, and it's just a way that your body's probably naturally doing this. Now, there are some people that think it's hormones in the diet and things like that, and there hasn't been very good evidence to suggest that's true, but nonetheless, it's possible. But most scientists think that you're tricking children today in the United States are tricking their body to think, hey, I've got all this stored up energy in the form of fat. I should go into maturation and start reproducing. I should start producing eggs or sperm. And it happens more in females because females, their gametes are much more energy rich than males. Now, males' sperm is really cheap, okay? Energetically speaking. It's very, very cheap. It doesn't cost very much to produce, so we probably won't see the trends so much in males. Now, males have slid down a little bit on the age for maturation, but are pretty stable. Females mature, a couple years before males now, on average. So, and if you want to know more about that, shoot me a line, and I can give you some papers on, that kind of trend. All right, so let's look at the stages of meiosis. Meiosis occurs in two different stages. There's two different main divisions. We have what we call meiosis 1 and meiosis 2. Now, I'm going to overview the whole thing real quick, and then next lecture I'm going to talk about meiosis 1 in detail, and then the lecture after that I'm going to talk about meiosis 2 in detail. So first, meiosis 1 separates those homologous chromosomes. So if you remember, this is very similar to mitosis, where the cell's going through the cell cycle. It goes through the G1 phase, goes into the S phase, copies all its DNA, goes into G2 phase, then it goes through the first M phase, meiosis in this case, where those homologs are separated. The homologous pairs are separated. Now, like I said before, that S phase occurs before the M phase. So DNA is replicated during the S phase, just like mitosis, before meiosis occurs. After the homologs are separated, then in meiosis 2, those sister chromatids are separated. So it's a reduction division. You're not just separating the sister chromatids. That's what happens in mitosis. In meiosis, you're separating homologs. So you either get dad's chromosome number 1 or mom's chromosome number 1. And then in meiosis 2, you're separating the copy of that. So you already inherited, dad's chromosome number 1 or mom's chromosome number 1, depending on the sperm or egg that you're looking at. And then you inherit just one copy of that, okay, which makes you a haploid in the case of humans. We'll go through this and you'll see what I'm talking about. So instead of at the end of mitosis, when we talked about mitosis where we went 1 cell to 2 cells, meiosis is 1 cell to 4 cells. Okay? And the 4 cells are going to be half of the original chromosome as that beginning cell. So here's kind of how that works. Germline cell, after the S phase and G2 phase and G1 phase, it's ready to replicate. So the DNA has already been replicated. The cell's big enough. The cytoplasm's there. Any mitochondria or anything else that's occurs in the cell, depending on the cell type, okay, or I should say, but depending on the individual and the organism that we're talking about, you might have quite a few organelles in here or very few organelles in here. Okay? But nonetheless, DNA has been copied. It goes through the same kind of system. Prophase, but now we call it prophase 1. Homologs line up. Meiotic spindle forms, okay, chromosomes condense, nuclear envelope goes away, goes into metaphase, homologous chromosomes line up on the metaphase plate, okay, spindle apparatus is formed, microtubules are attached to the chromosomes, and then those homologs, the homologous chromosomes, are separated, okay, anaphase 1. Telophase 1, homologs, new nuclear envelope forms around them real quick like, pinches off, okay, and then the system just keeps going. There is sometimes a G1 phase right after this, just to install a little bit of more growth in these cells before they go and replicate again, but there is no S phase, so you're not copying the DNA again. Right? And it depends on the organism. Sometimes it just goes right in to prophase 2. So telophase, prophase 2, and cytokinesis would happen here. It would split the two cells, or make a plate to split the two cells. And then so it goes into prophase 2. Again, the chromosomes condense, meiotic spindles form, nuclear envelope goes away. Metaphase 2, homologous chromosomes line up on the metaphase plate, spindles attached to them, etc. Now, instead of, I'm sorry, not homologous chromosomes, just chromosomes. My bad on that one. And now those chromosomes, which consist of sister chromatids, are separated. And at the end, it pinches off, and now you have 4 cells from the original 1 cell. And in this case, this was a diploid individual producing 4 haploid gametes.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only one of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of four genetically different haploid cells.
Okay, so that's the overview of meiosis I and meiosis II. Now, they talked about a unique event in meiosis called crossing over, and we're going to spend a lot more time about how this occurs. Again, sister chromatids exchange DNA material between the chromosomes, or the chromatids, sorry, resulting in what we call hybrid chromosomes. So at the end, there's this hybridization. So you get, normally you would be getting either your dad's chromosome number 1 or your mom's chromosome number 1. But because of crossing over, you get part of your dad's chromosome and part of your mom's chromosome. Or you have the capability to have that, or possibility of having that. The other thing is, like I said before, you either get chromosome number 1 from your father or chromosome number 1 from your mother. Okay? And because of that either piece, we call that independent assortment. And it doesn't, because you got chromosome number 1 from your dad, doesn't mean that you're going to get chromosome number 2 from your mom. You could get chromosome number 1 from dad, number 2 from dad, number 3, number 4, all the way, the whole way. You get every single chromosome from dad. It's not likely, but then again, even if that occurred, crossing over is most likely going to give you some of mom's chromosomes, some pieces of mom's chromosomes. Okay? And so we'll look at the likelihood of this and how many possible combinations you could expect from things like independent assortment. Okay? So next time I'm going to spend a lot more time talking about meiosis I, or that initial separation of homologous chromosomes.